Welcome to SNEV Podcast.
Hello everyone and welcome to another episode of the SNEV Podcast. Today we'll be talking to Guillaume Van Niel about some of his work in EV intercellular communication, EV cell biology, and the quite influential review he wrote on that topic. So just to start off, Guillaume, do you want to introduce yourself and maybe talk a little bit about how you got into EV research in the first place?
Yeah. Well, hello everyone. Thank you, Liam, for the invitation. So, if I have to start about my first steps into the EV field, we have to go back to the last century. So it started in 1998 when I applied for an internship for my master, and I was lucky enough to meet a fantastic supervisor, Martine Eman, at the Necker Hospital, and she had the kind of puzzling story. She was working with intestinal epithelial cells, and each time she was feeding the cells with antigens, on the basolateral side there was peptides of 8-12 amino acids constantly being recovered without being degraded. And she was wondering why we still have these peptides of this length on the basolateral sides, and her postdoc, Kathleen Terpan, heard about a young scientist, Clotilde Terry, talking about extracellular vesicles with MHC class 2, MHC class 1 at their surface, and just proposed a kind of crazy idea. What if intestinal epithelial cells would produce extracellular vesicles harboring MHC class 2 that would be accommodated with these peptides? And that's how everything started. So, well, in 1998, nobody was working on EVs. We were all talking about exosomes, by the way. And that was the start of a great adventure that I'm still continuing right now.
That's really interesting. So you've been around at the very beginning. What exactly was that like, in those sort of formative years where ISEV first started to come together?
But it was, a bit, it was very exciting because we were very few and kind of secret community working on something nobody have heard already. So we had some nickname like proctolog of the cell, and people did not believe much into EVs at that time. And when I defend my PhD, maybe one of the first PhD on extracellular vesicles, in the references there was 17 paper about extracellular vesicles published so far.
Wow, only 17?
17, yeah. So we had to convince people that we were not just working on garbages, and that there was this new field opening. And I was lucky enough during my master to collaborate directly with Graça Raposo, and then also during my PhD. So, well, I had two great godmothers with me, helping me to go through the EV field.
Yes, that certainly would be very helpful. So what were the sort of steps from that point onwards? What was it, around 2010 that the first ISEV meeting took place, right?
Mm-hmm.
What was it like then during the, when that first meeting and when, the term EV was sort of first coined and proposed?
It was super friendly, I have to say, because it's like when a new topic is emerging, you have only passionate people in the field happy to share and to build something. And there is not so much competition. We were all coming from different fields of research and rather trying to help each other than trying to copy each other and to compete. So, that was a super atmosphere.
You also mentioned how it felt like you were onto something secret at the time. Was the fact that no one was really talking about this too much, or people didn't really think so much of EVs or exosomes. Was there a sense that you guys were just onto something that was inevitably going to be so big and, so impactful?
Well, we had no idea that it would be so big at that time, because you have to keep in mind that the main paper at that time were about the clearance mechanisms that I think are still super important. And then with a paper of Grassa-Raposo about capacity to signal with recipient cells, and then the next papers from Jan Lodval about transfer of genetic materials, then it opened a brand new field with a lot of opportunities. But I would say that at the very beginning, we had the feeling that we are touching a new way of communication, but we had no idea about the, let's say, how many different pathways and processes that will be involved in, and how we could translate that into clinical trials at that time.
And I suppose it must have been much easier to, wrangle consensus at the time when the community was a lot smaller.
Well, consensus, not so much. I mean, everybody was coming up with a new name. Exosomes, tolerosomes, oncosomes, and there was a huge work made, notably by the ISEV, by the ISEV community, to really shape this nomenclature. So we were all talking about the same thing, the same way of isolating them and characterizing them. So to be sure that we were talking about the same thing.
Yeah, it sounds like it must have been a very exciting time. It's funny you should say that you got your start doing an internship before your master's, because I did much the same thing. I did like a 4-week scholarship at Andy Hill's lab, and that was where I first learnt about EVs. And I can remember when I first went into that lab, they gave me these 2 papers and told me to go over them quite thoroughly, just to learn about basic EV biology. One of them was ISEV 2018, and the other one was your review, the 2018 article titled Shedding Light on the Cell Biology of Extracellular Vesicles. And now, in retrospect, considering that is one of the most highly cited reviews in the EV field, we could say that it's, at the time at least, was, probably the foremost distillation of knowledge we had on, the cell biology of EVs. It was very well done, it was very clear. And so could you maybe give us a little bit of insight on how that review came to be, and maybe a few pointers on what you think it might take to write a successful review?
Yeah, well, thank you first. That's well appreciated. Well, this review, that was the idea of Grassa-Raposo, where I was as a researcher at that time, and yeah, we are not so many people are really focusing on the cell biology. So all this kind of complex molecular intracellular interaction and dynamic that leads to the biogenesis of extracellular vesicles. People are more into what their function and what can they do and how can we translate that into clinics. But what I always believe is that you need fundamental basis to understand what you're working with, and how you can then really advance our knowledge about the EV field. And I was already in the field since a long time, and I was working on EVs, but I did also one postdoctoral one postdoc with Willem Sturvogel in Netherlands, where I did a true cell biology, biochemistry on ubiquitination. In the lab of Grassa, I was working also on the sorting mechanisms about MVBs, in MVBs. So what I did first was to take all the slides I had, where I'm doing a lot of drawings. So I started with the figures, because that's how I work. I look first at the figures of a review, and that gives me really a good overview about what I'm going to read afterwards. but I really need this visual support to understand things. So that's how also I try to transmit what I know. Start with that, and well, maybe people are thinking my figures are a bit too complex. I've got this kind of also of remarks for the last nature review we did about the challenge in the EV field with Dave Carter notably and Peter Andrasa. But especially when it comes to cell biology and intracellular trafficking, it's much easier to make a drawing than a list of molecular interactors and how they, in a chronologically order, works and interact, etc.
Yeah, certainly. A picture says 1,000 words. So the function of a review is to take a whole lot of ideas or concepts and sort of stitch them together, sort of smooth out the edges and paint a coherent picture, right? And again, that's what you did in the review, and that's what you're doing with these figures. So do you maybe have a sense that at the time some of these ideas that you connected together weren't perhaps quite as well connected as they could have been, and maybe that's why the review was so successful? Do you think maybe there was a little bit of a gap there that, maybe some of these ideas at the time weren't as well stitched together as they could have been?
That I don't know. I don't know if I proposed new ideas. I think I just digest everything I could read and into a kind of cohesive picture of what can be proposed, and without taking into account also the cell types. Although each cell type has its own specific organization, I think there are general rules we can propose for the EV biogenesis. This is, by the way, what we try to do while we are writing a new review, but now in recipient cells, so using exactly the same approach, so trying to give a global view of the cell biology in recipient cells of extracellular vesicles.
Yeah, I saw that more recent review you've written is on intercellular communication.
No, I'm talking about a new one.
Oh, right. Yep, I see. I get you. In that case, what ways would you say our understanding has sort of changed or progressed most dramatically since that time?
But what do you mean exactly in terms of what? Of biogenesis pathway, or about the function of EVs, or?
Yeah, I suppose in terms of cell biology, intercellular communication, any of these areas, and in what ways do you think our understanding's changed the most?
Well, the thing is that it became much more complex than at the very first work of Harald Stenmark or Mika Simmons, and we don't have only escort machinery and escort independent, and we have many more mechanisms because we have many more EV subpopulation. If you take the work of Edith Buzas, for example, about secretory autophagy, if you look at large oncosome, if each time you look at a new type of EVs, like the EVs that are budding from the tip of the cilia, each time you have kind of a new mechanisms of biogenesis. So I would say that's one thing is the complexity. Then the second thing, and it's something I really try to, a message that I try to transmit is how do we see this biogenesis? Is it an action on purpose? So is it really something that the producing cells is doing to communicate? Or is it something that the cell is just doing for her own benefit without taking into account any kind of communication? And that's a kind of philosophical question. So are EVs part of a true dialogue, or is it two parallel monologue between one cell that produces for our own benefit and one cell that recover EVs for our own benefit as well?
Yeah, that's really interesting.
Without really a communication established between both cell types.
So do you explore this sort of more philosophical question or anything like it in this new review?
Yeah, it's always the kind of question I have is everybody is excited by, well, my cells are producing EVs for communicating with these other cells. But, well, first, what's going on in the producing cells? And how do you know that also in vivo, this EV will be directly targeted to the recipient cells for this purpose? So far, we don't have good evidence for that.
So I can see the name of your current research group is EV intercellular communication and their application in therapeutics, or EV-CAT. Are these the...
Yeah.
Does that title just about summarize your research interests at the moment?
Well, what we do is that we explore further the cell biology. So, but at the same time, we are trying to harness this fundamental knowledge to really try to manipulate the different step of the EV life cycle. For example, we have tools, autogenetic tools to stimulate EV secretion. We have chimeric receptor to capture them. We are developing new tools to follow delivery, if there is any delivery. So these kind of things, and then we use the zebrafish to really assess the therapeutic potential of these EVs. So it's really starting from fundamental cell biology up to, well, preclinical assay.
Yeah, the zebrafish models are really interesting. Do you want to maybe share a bit more about how those are helpful for EV research and give a bit of insight for those who aren't too familiar with them?
Yeah, well, I have to say that everything started with the zebrafish with Frédéric Verveil. We met also quite a long time ago, and we had the same idea. He had developed this fantastic CD63 fluorine EV reporter, and one idea I had was to transpose it in zebrafish. One idea he had was to use zebrafish for his tool, so we kind of agree on a common program. We are both with Fred using a lot of imaging to try to prove our concept and to prove the mechanisms that we want to explore. And for us, the zebrafish had this advantage of being transparent and super small, so we could put it under a microscope and do in vivo cell biology. So that was the main approach. The second approach was really that we had the surprise to finally see EVs in the blood flow. We were not expecting that because the tool that Fred had developed at that time was really to see the fusion of MVB with the plasma membrane. But when he came back with movies of EV flowing in the blood circulation, we said, okay, we have something big here. We really have to focus on that, forget about MVB secretion, go on the EVs, track them, see where they go and what they can do. And well, since that, well, we are both him in his new lab in Utrecht, and myself, we are trying to capitalizing on this kind of discovery. And for me, the zebrafish is really a complementary model that really bridge in vitro study and everything we can do with cell culture, with imaging, and in vivo study with mouse, where you cannot do this kind of imaging, at least at this spatiotemporal resolution, at the scale of a single EV, and really in live in all kind of tissue.
So what do we know and what don't we know about biodistribution at the moment? You know, what have we learned in a zebrafish that does translate to, you know, a mouse or a human, for example?
The good thing with the zebrafish, that is maybe also a disadvantage, is that we can see EVs, so you need to label them. And the first thing that you learn is, depending on the way you label your EVs, whether with endogenous labeling approaches or post-isolation with all kinds of dyes, the results are different. And the main problem is then to know what you are tracking. Is it the EV or is it the dye? Yeah, so there was a really recent paper stating that PKH is not a good dye to follow EVs, notably through the blood-brain barrier. I think that this kind of paper is as important as paper claiming a new role for EVs. If you know what you should avoid in terms of methodology, that's super important for your way to build your new project. And what we have seen so far is that at least under physiological state, there is not so much crossing of any kind of biological barrier. I don't say that it does not exist. I just say that with the type of EV that we are labeling, with the site of production, with the cell types we are using, they do not cross. That doesn't say they never cross. You have a lot of pathological conditions where the biological barriers are permeable, notably during inflammation. So they could do. Now, whether there is a specific mechanism of transcytosis allowing EVs, for example, to reach the brain or to come from the brain into the general circulation, that's the kind of proof I'm still waiting for.
So considering intercellular communication and the transport of EVs, what are the sort of big questions that remain to be answered, aside from, the crossing of biological membranes?
One first question will be the range of action. Where do they act? Do they act really on the cells that are producing them? Are they just acting on the neighboring cells? And of course, if they are directly produced in the blood circulation or delivered into the blood circulation, up to which organ they can go, and if they are in particular in the blood circulation, what is their half-life and their clearance? And finally, what amount of EVs is able to reach a given organ. That are part of the main question about the biodistribution, I would say. But it's all about in vivo, in fact.
Yeah, exactly. These are the sort of questions that you can't really answer so easily in vitro. So are there any of these questions that you think are perhaps being held back by, some technological barriers that need to be overcome? Maybe we need more sensitivity or specificity. Are there any new technologies on the horizon that you're particularly excited about that might answer some of these questions?
New technology. I would say if we are able to really be sensitive enough to detect a little amount of EVs in a given organ. So I'm talking rather about analysis on a chip or a sorting device able to really isolate subpopulation of EVs, then that should be a very important step. There is always this sentence of Clotilde Thierry that I like to quote, saying that the EV field is at the same state of understanding and development than the immunology and white blood cell field in the 50s, meaning that we have, and I think she's perfectly right about that, we have a lot of subpopulation of EVs now, but we always consider just one population of EV doing everything. And I don't think that's true. I think it's like with T cells, macrophages, neutrophils, and so on. Each subpopulation of cell has a given purpose and role, Whether a subpopulation of EVs, it's just a clearance mechanism, whether it's involved in a kind of communication with another recipient cells. But if we don't have a precise repertoire of all EV subpopulation, their composition and what they can do, we are still stuck at global view and really in a blurry situation.
Oh, that's a very pragmatic take. It's also probably a very accurate and healthy one. So that's a good segue into the EMBO practical workshop. Do you want to talk a little bit about what happens at this workshop that you've been helping to organize, and perhaps, talk a little bit about the value of these kinds of workshops?
Yeah. Well, I have to say I am involved now as organizer, but everything started with Anne Hendricks and SN Toon, and they invited me as an electron microscopist expert the first year, and then I got involved in the organization, and now Cecilia Lasser joined us. The thing was that at that time, so we have started that in 2014, I think. And there was no way to learn the basic tech to isolate, to characterize EVs. So that was the first idea, the basic idea. Let's teach this new generation of students how to properly isolate EVs and to teach them about this commercial product like ExoQuick or other precipitation assay, how they can be misleading in your interpretation, and what are the main way to characterize EV, how we can draw a conclusion from our essay. And now we are, I think it's the 10th edition this year. So it's quite a successful course. I mean, we have only 24 students selected, but we had more than 200 applications again this year. And we have a kind of game now that is to divide the 24 students in 3 groups, and each group has a sample where we have spiked either lipoparticles or referent EV, and they have to find out, after isolating this EV and characterize them with different technology like super resolution and what do we do, flow cytometry, electron microscopy, to find out what kind of sample they have. So it's a game and it's a course at the same time, and it's pretty heavy because they have also to present their data, they have to present their own research, but it's a lot of fun, and I have to say that the environment, the EMBL in Heidelberg, is a fantastic place to go and to organize this kind of workshop.
So who are these workshops sort of targeted at?
What do you mean?
Oh, the practical course, sort of. Who is the target audience? Is it new researchers or?
The target, it can be anyone. I mean, we have master students just entering in the field and quite new. We have PhD students and postdoctoral fellow, even sometimes some team leaders or PIs. And we try really to mix people with no experience, people with a bit of experience, to really have a kind of dynamic into the working group that we have. And we have always a good support also from the trainers that are most of the time the students of our lab who are helping us to do the practice, and also the MBL support members. But it's open to everyone. I mean, you can apply if you want.
Yeah, no, it sounds like a really good course. It's very good that the EV field has this sort of thing. So considering this is a student podcast, just for the final question, do you have any sort of tips for, maybe early career researchers or, people who are just starting out in the EV field, career advice, anything like that?
If they want to stay in the EV field, you mean?
Yeah, that, or, for anyone looking to enter the field.
Well, I don't know if I have any tips. I know what I did. I didn't follow the mainstream. And I would advise people to do the same. I mean, to try to read a lot, to have a global idea about what's going on, and then to find an idea or a concept or a pathology where nothing has been explored in terms of EVs. That would be my main tip, I would say. But it can be methodology, it can be clinical relevance, it can be cell biology. But you have to find your own identity by developing your own ideas.
Yeah, I suppose if, as you said, the EV field is where the immunology field was 50 years ago, then there are going to be no shortage of these little, questions and areas that need to be answered. So, so yeah, thank you. I think there's a lot of interesting stuff here for our listeners. Thank you again for talking to us today, Guillaume.
Thank you.
So that has been this month's SNEV podcast. Again, my name is Liam Hourigan, and we hope to catch you next time. Bye.